This is DNA replication by Kimberly Curris. Before we get into the actual process, we must first understand the key enzymes that go into DNA replication. Helicase unwinds the DNA strands at each replication fork. Single-stranded binding proteins, or SSBPs, bind to the DNA to keep the strands open and prevent rebonding. Topoisomerase prevents strain ahead of the replication fork by relaxing supercoiling. DNA polymerase III attaches to each strand's primer and moves from 3' to 5' as it adds a new strand of nucleotides in 5' to 3'. RNA primase initiates replication by adding short segments of RNA called primers to parental strands. DNA ligase fills in the gaps and connects Okazaki fragments on the lagging strand. DNA polymerase I replaces RNA nucleotides from primers with DNA nucleotides. Now that we understand all the enzymes of DNA replication, we can begin dissecting the process of replication. Once DNA is ready to be replicated, helicase comes in and begins to separate and unwind the two strands. As it does this, topoisomerase attaches to the DNA ahead of the helicase to prevent strain as the strand unwinds. Next, SSBPs attach to each strand, helping signify the replication fork and prevent rebinding. Now that the two strands are fixed apart, RNA primase inserts 3 RNA nucleotides to the start of each DNA strand. Once the primers are inserted, DNA polymerase III comes in and begins adding complementary nucleotides starting at the primer. DNA polymerase moves from 3' to 5' so it can add nucleotides in one continuous period on the leading strand as it follows helicase. I'm showing the synthesis of the leading strand first and then the lagging strand, but keep in mind that these processes are simultaneous. Onto the lagging strand, RNA primase has to add another primer since DNA polymerase can only move in the 3' to 5' direction, which is opposite to the lagging strand. DNA polymerase III starts at this primer and adds new nucleotides until it reaches another primer or the end of the strand. RNA primase has to continue adding more primers, and this process repeats until the whole strand is synthesized. Once both of the strands have been replicated, DNA polymerase I replaces the primer's RNA nucleotides with complementary DNA nucleotides. Since the lagging strand can't be synthesized in one continuous movement, the new strand is synthesized in sections called Okazaki fragments. DNA ligase must come in and attach these fragments together and fill in any holes that may have been left by DNA polymerase III. Newly synthesized semi-conservative DNA winds into double helixes with the parental strand in green and the new strand in blue, and that's DNA replication. Thanks for watching!